I'm Cody, and I'll be presenting redox and serpentinization conditions in geology. This is sort of a couple of topics that I'm interested in that I thought I would share. So the two topics, one being the redox conditions, determining ferric-ferrous ratio in minerals, with the hope of being able to determine oxygen fugacity in magmatic systems so that you can ultimately figure out what the oxygen budget is of a system and potentially even the oxygen fugacity history, because X-ray absorption spectroscopy is a probing technique. It only needs very small sample volumes, and you can even probe individual points within a crystal. So potentially the closure temperature of that zone. The other topic I'm interested in is something I did during my dissertation, which is relating serpentinization environment to serpentine crystallography through characterizing the very details of the crystal structures of serpentine minerals. So the main thing with the X-ray absorption spectroscopy that we're working on is figuring out this anisotropy problem. Because in any type of absorption spectroscopy, if you're going to get concentration of something due to an absorber, you must get total absorption, total absorbance. And in an anisotropic material, you have very different absorption magnitudes with orientation. So you have to actually kind of calculate what total absorbance is. So to analogize with visible light, this is biotite. If you've ever seen under a light microscope, Pleochroism, in one orientation, a biotite crystal absorbs a lot of light, so it appears black. And if you rotate the stage, it transmits light. So that's just Pleochroism. This image is monochromatic light, though. And if you plot out absorption with orientation radially on the right, this is what it looks like. The absorption magnitude has this sort of cosine square shape, and that's characteristic of individual crystals. So the same thing applies to the infrared wavelengths. This is well known. The only difference is these are molecular vibrations that are absorbing the incoming radiation. This is much broader wavelengths of electromagnetic waves. But as we've seen with X-rays, it's the same concept carries through. You're just at much shorter wavelengths. And also there are some minute differences due to local structure that arise with X-rays. But overall, it has this same sort of shape, the same absorption shape. So we're applying this principle, and X-ray wavelengths are sensitive to this ferric-ferrous ratio. So that's why we're doing this. And this is just demonstrating that, like, we can orient crystals in different ways and analyze a crystal in a specific orientation when needed. To get the absolute characteristic absorption of a crystal in different orientations. And this is showing an X-ray absorption spectra in different orientations, just from 0 to 90 degrees, from normal to the B-axis of a pyroxene. On the y-axis is absorption. It's just arbitrary units because everything has to be normalized. And on the x-axis is energy. But as you can see, yeah, from one orientation to another, you have very different absorption spectra. The other topic I'm interested in, though, is serpentinization environments. This is the hydration of ultramafic rocks under green schist to amphibolite facies. And this has applications in planetary because you're dealing with a lot of ultramafic compositions like meteorites and mafic compositions as well, if you're talking about the surface of the moon or Mars. And if that material is hydrated under certain conditions, you form serpentine minerals or clays. And these are very interesting in geology as well because they relate to the origin of life on Earth. This is one of the scenarios that's suspected because there are reactions that are associated with those that produce amino acids. But I'm just interested in these because serpentine minerals are very difficult to solve their structures, but they do reflect their geologic environment, at the very least on a broad level. But I believe that you can actually really constrain the environment with these minor differences in structures. This is just a map of Idaho. There's some green dots on here, just showing different locations that I went through. So I did my this dissertation chapter on western Idaho's serpentinites, which is a good location because you have this swath of ultramafic compositions that have experienced different metamorphic conditions from west to east. On the western side, you have lower temperature ultramafic rocks, and which are like still preserve their oceanic origin. And as you approach continental Laurentia, you have this suture zone where the metamorphic grade is higher. So you have higher temperature serpentinization, and maybe even higher than that, where serpentinization doesn't occur. In the upper left, this is just going from the outcrop scale down to the nanoscale is sort of my favorite thing to do. These are very heterogeneous usually because they're unstable and crustal rocks and form things like talc along their contacts. And I can probe different zones or collect rocks from different zones in a rock and then look at a particular texture at the microscale in the lower left, and then look at that texture at the nanoscale, or pick textures to look at the nanoscale, this being chrysotile asbestos in this case. There's also a broader impact of this being the fact that these are associated with the development of asbestos. So that's being an environmental hazard, but also because of those metasomatic zones, these things just slide into the highway. They're a huge landslide risk anywhere that they occur. This one just happened like 3 weeks ago at the outcrop that I just showed in the last slide because of those metasomatic zones. But the primary two methods that I use for characterizing these is high resolution transmission electron microscopy and single crystal X-ray diffraction. The cool thing about the TEM though is that we can directly, we can collect an image, get chemistry and electron diffraction at a very fine scale. We can prepare like a thin section, it's a special thin section, and probe specific textures within a rock to get these. We can basically image atoms almost, you know. And the X-ray crystallography just for the absolute structural determination of these. This is just an example of some photos of the TEM textures. In this case, this being a clinochlore with an epitaxial overgrowth of two-layer antigerite. It's just a specific type of serpentine that occurs probably at very specific temperatures. It's a rare polytype. At the nanoscale in the upper left, all the black and white photos are at the nanoscale. You can see the very planar structure of clinochlore, and then the wavy structure part is the antigerite, which is a modulated structure. And the basic idea here being that the spinel structure is what usually has aluminum at higher temperatures. You go down to lower temperatures, retrogress it, the aluminum goes to clinochlore and apparently this two-layer polytype. So it's reflecting the environment and chemistry, in other words. And this structure had never been solved before. This is a video. I don't know if it's going to work, but this is just a video. If you click play on here, of the structural solution. As I mentioned, they're not periodic, or they're not very periodic in 3 dimensions, so you have to solve them in higher dimensions, basically. But yeah, that's it.